Greetings to one and all. This is Dr. Sanjay, Professor of Physiology, Chettinad Hospital and Research Institute, Chennai. I will be updating you on sleep, its core principles, its architecture and functional significance in this session. These are the objectives of my session. I will begin the session talking to you about the core principles of sleep physiology, following which I will discuss the architecture of sleep, under which I will talk to you about the sleep pathways which will help us to understand the neural regulation of sleep better. And then I will talk to you about the two types of physiological sleep, namely NREM sleep and REM sleep. And then I will talk to you about the significance of the physiological variations during sleep, after which we will discuss the functions of sleep. I will conclude the session giving you an overview of the diagnostic intervention of sleep, namely polysomnography, and then I will talk to you about the consequences of sleep deprivation. And so let us begin to have an update on the core principles of sleep physiology. There have been many definitions of sleep in the literature, but a simple definition is sleep has been defined as a physiological state of relative unconsciousness with inaction of voluntary muscle effort, the need for which recurs periodically. Sleep has been classified based on its functional depth as light sleep and deep sleep, based on its electroencephalographic characteristics as desynchronized sleep and synchronized sleep, and based on its physiological characteristics, namely non-rapid eye movement sleep or NREM sleep and rapid eye movement sleep. Now let us discuss the historical milestones in the physiology of sleep. It was in the 17th and 18th century that the father of medicine, Hippocrates, along with Galen, proposed that sleep involved the brain and was as important for homeostasis. And so in the 19th century, sleep researchers began to record brain activity and notice changes during sleep. The 20th century saw quite a few advances in the physiology of sleep, following the invention of the electroencephalogram by Hans Berger. Nathaniel Clayton made landmark discoveries on the physiology of sleep during the early 20th century, and it was in the mid-20th century that REM sleep and non-REM sleep were discovered. The late 20th century saw development in the neurochemistry of sleep. In the current era, that is the 21st century, quite a few advances on the physiology of sleep have been discovered. With the advent of radiological diagnosis such as fMRI and the PET scan, the plasticity or how brain networks change during sleep have been discovered. Sleep's role in memory consolidation, immune function, metabolism and brain waste clearance by the lymphatic system became evident, giving to us a better understanding of the functions of sleep. And the molecular mechanisms which control sleep have also been discovered by different genetic studies, which is an area of ongoing research in the physiology of sleep. Recognition of sleep disorders such as insomnia, narcolepsy, sleep apnea improved their understanding and treatment. Crescendo sleep refers to normal sleep during which there are gradual movements of the sleeper during the course of sleep. The two physiological types of sleep are NREM sleep or non-rapid eye movement sleep and REM sleep or rapid eye movement sleep. This is based on the movements of the eyeball during the period of sleep. Functional sleep is graded based on changes noted in the electroencephalogram or the EEG. Since the electroencephalogram of REM sleep is similar to that of wakefulness, REM sleep is also known as paradoxical sleep. Dreams are significantly noted to take place during REM sleep, but also occur during NREM sleep. Now let us try to understand the normal sleep cycle. The sleep stages appear in a predictable sequence which repeats about every 90 to 100 minutes. In a typical night in a young adult, after the lights are turned out, there is a period of wakefulness before sleep appears, and the duration of this period of wakefulness is known as sleep latency. Then one enters the NREM sleep, and after about 1.5 hours or 90 minutes, REM sleep appears. The NREM-REM cycle goes on throughout the night, and a typical night may contain 4 to 6 such cycles. This diagram shows a normal sleep cycle. The shaded area here refers to the NREM sleep, which alternates between the unshaded area, which is the REM sleep. And this keeps repeating itself, and in a typical night, there are about 4 to 5 such cycles. This slide shows that sleep patterns vary during different stages of development. Here you can see that human fetuses sleep most of the time, mostly exhibiting the REM type of sleep. At birth, newborn babies sleep for about 18 hours, 50% of which is occupied by the REM pattern of sleep. In adulthood, between the ages 20 to 70, most of the sleep cycle is occupied by the NREM pattern of sleep, the REM pattern accounting for only about 25% of the sleep cycle. It is interesting to note that sleep deprivation is associated with longer periods of NREM sleep. A knowledge of the waveforms of a normal electroencephalogram is mandatory to the understanding of the physiological sleep cycle. A normal electroencephalogram shows 4 types of waves, namely the beta wave, the alpha wave, the theta wave and the delta wave. These 4 waves are based on their amplitude and their frequency. The beta wave occurs when a person is awake and thinking, and this is how the beta wave appears. The alpha wave occurs in a relaxed and awake person with his eyes closed. And the theta wave occurs during light sleep and the delta wave occurs during deep sleep. We will come to more on the EEG findings during sleep later in the presentation. Now let us try to understand the differences between desynchronized sleep and synchronized sleep. This classification is based on the EEG characteristics noted during the sleep period. Desynchronized sleep refers to REM sleep. Here you have the EEG showing low amplitude and high frequency waves. This EEG pattern is also noted to occur during wakefulness. Synchronized sleep refers to NREM sleep, during which the EEG shows high amplitude and low frequency waves. Here you have the EEG tracings showing the desynchronized sleep or the REM sleep, where the frequency of the waves are increased and the amplitude decreased. And here you have the NREM sleep, where the frequency is decreased and the amplitude is increased. That is the synchronized type of sleep. Now let us take a look at the currently accepted theory of sleep. Sleep is caused by an active inhibitory process. This is what is believed as on date. An earlier theory of sleep proposed that the excitatory areas of the upper brainstem and the reticular activating system to become fatigued during the waking day and become inactive as a result. It was later discovered by an experiment that transecting the brainstem at the level of the midpoints created a brain cortex that never goes to sleep. This experiment facilitated the current view that sleep is caused by an active inhibitory process whereby a center located below the midpoints of the brainstem appears to be required to cause sleep by inhibiting other parts of the brain. Now let us take a look at the genetic modulation of sleep. This slide shows 3 categories of genes with a few examples in each category. You have the core clock genes which regulate the circadian rhythm, and the genes which influence the sleep duration and quality, and the genes which are associated with the disorders of sleep. This slide gives you an overview of sleep in the animal kingdom. It is interesting to note that cows sleep only for about 4 hours in a 24-hour period. And here you can see the bats and the koala bears sleep most of their lifetime, that is 18 to 20 hours per day. As far as humans are concerned, the functional sleep duration in an adult human is about 7 to 9 hours, and the sleep duration in infants, as I discussed earlier, is about 16 hours per day. Having gone through the core principles of sleep physiology, let us now open our eyes on the architecture of sleep. Here we shall discuss the sleep pathways which will help us to understand the neural regulation of sleep better. And then also let us take a look at the 2 physiological types of sleep, namely non-rapid eye movement sleep and rapid eye movement sleep. This slide gives you the basic architecture of the sleep pathways, and here you can see that wakefulness is mediated by neurons of the reticular formation of the brainstem, and these neurons project to 2 regions, namely the cerebral cortex and the thalamus. With regards to the pathway projecting to the thalamus, there are 2 groups of neurons, namely the histaminergic neurons and the cholinergic neurons. The cholinergic neurons are also known as wake-on REM on cells because they are active during wakefulness and REM sleep, and they become inactive only during NREM sleep. Now there is another group of neurons known as the orexigenic neurons. These are neurons which mediate the sleep-wake cycle. They are located in the hypothalamus and they use a neuropeptide orexin. They are excitatory to the neurons related to the sleep-wake cycle. These neurons are active during the wakeful state and during REM sleep. These groups of neurons play a critical role in the overall regulation of sleep. Having gone through the sleep pathways, let us take a look at NREM type of sleep. Non-rapid eye movement sleep is also known as slow wave sleep or synchronized sleep, because as we saw previously, in this type of sleep, the frequency of brain waves are much slower when in comparison with REM type of sleep. In functionally fit individuals, sleep begins with the NREM type of sleep. It is a restful type of sleep in which the individual is relieved from the tasks that he or she is normally subject to. Normally, NREM type of sleep shows 4 stages, and stage 1 you have the low amplitude mixed frequency waves. Stage 2 you have waves characterized by sleep spindles and K complexes. Stage 3 you have high amplitude and the slow theta waves, and stage 4 you have high amplitude and the slow delta waves. This can be seen in the tracing. Here you can see stage 1 you have low amplitude mixed frequency waves. Stage 2 you have the K complexes and the sleep spindles. Stage 3 you have high amplitude and the slow theta waves, and stage 4 you have high amplitude and the slow delta waves. REM sleep, on the other hand, is also known as fast-wave sleep or desynchronized sleep or paradoxical sleep or dream sleep or deeper sleep. During REM sleep, the EEG is characterized by high frequency, low amplitude pattern, and this type of a pattern is also seen during wakefulness. In cats, REM sleep is characterized by a type of waves known as the PGO waves or the ponto-geniculo-occipital waves. PGO waves are not seen in humans by scalp EEG recordings, but they are recorded in in-depth EEG recordings. Now let us take a look at the physiological changes noted in NREM and REM sleep. The eye movements are slow and rolling in NREM sleep, and they tend to disappear in stage 4 of NREM sleep. As far as REM sleep is concerned, the eye movements are rapid, and that is why you have the term rapid eye movement sleep. The muscle tone decreases progressively in NREM sleep, and in REM sleep it reduces to a state of hypotonia. Heart rate, blood pressure and respiration decrease in NREM sleep, and the heart rate and respiration are irregular in REM sleep, and the blood pressure is variable in REM sleep. Body metabolism slows down in NREM sleep and begins to rise in REM sleep. Now coming to the other physiological changes, we will be seeing later on, growth hormone and gonadotropin secretion by the pituitary gland increases during NREM sleep, and as far as REM sleep, there are three characteristics which are noted, namely twitching of the limb musculature, penile erection in males, and engorgement of the clitoris in females, teeth grinding or bruxism in children. Let us take a look at a few behavioral changes of NREM and REM sleep. In NREM sleep, there is a gradual decline of consciousness, the sleeper showing a graded increase in resistance to being awakened. It is almost difficult to arouse a person in REM sleep, and you can say that REM sleep is a state of relative unconsciousness. There is no recall of dreams in NREM sleep. There is vivid recall of dreams in REM sleep, and most of the dreams which we remember take place during REM sleep. The auditory reaction time becomes longer in NREM sleep. The visual response time to stimuli is variable. Thoughts become illogical and incoherent, and there is retrograde amnesia, which can be seen with the person not being able to recall the onset of sleep or not being able to recall dreams. Memory for the few minutes before sleep is also lost. Now we shall go on to see the significance of the physiological variations during sleep before going on to describe the functions of sleep. With regards to the functions of sleep, evidence-based research has been carried out on the following domains, namely ventilatory function, cardiovascular function, endocrine function, digestive function, sexual function, thermoregulation and cerebral function. One must note that disruption of vital functions during sleep can trigger pathological states and also alter the sleep-wake cycle. Now we shall go through the ventilatory functions during sleep, and you can see that the regulation of respiration varies during the states of sleep and during wakefulness. This can be graded as metabolic regulation and behavioral regulation. Metabolic regulation is what you would have studied in your textbooks of physiology as a neural regulation and chemical regulation of respiration, whereby the O2, CO2 and the pH levels are maintained in homeostatic ranges. Behavioral ventilatory control is mediated by the diaphragm, whereby a person can modulate his respiratory reserve voluntarily. A marked prevalence of respiratory disorders during sleep has been evidenced till date. Ventilation control is principally under behavioral command, which ensures a state of wakefulness. The onset of sleep inhibits the behavioral ventilation control. Thus, the shift from wakefulness to sleep coincides with diminished ventilation. Metabolic control gradually takes command of ventilation, but only becomes effective when sleep stabilizes. As a result, you have a state of periodic breathing where there is irregularity in the rhythm of respiration, and this continues till the metabolic control fully takes over. The period between wakefulness and stage 2 of NREM sleep is thus characterized by respiratory instability. And it is only during stages 2, 3, 4 and 4 of NREM sleep that the metabolic regulation of respiration confers stability to respiration. In REM sleep, ventilation decreases, but it's particularly unstable during this type of sleep. Age and sex have no significant differences in ventilation and in general regulation. Ventilatory dysfunction during sleep involves periods of awakening in order to ensure satisfactory ventilatory or respiratory homeostasis and the survival of the individual. With regards to cardiovascular function during sleep, bradycardia occurs during REM sleep and deep NREM sleep, and this is attributed to vagal activation and decreased sympathetic tone. Systemic arterial blood pressure diminishes during NREM sleep, especially in deep NREM sleep. This is related to peripheral vasodilatation and bradycardia. In REM sleep, contrary to NREM sleep, there is a wide variability in arterial pressure with upsurges of pressure simultaneous with rapid eye movements. The main changes in arterial pressure during NREM and REM sleep are related to the reduction in overall peripheral vascular resistance related to the reduction in sympathetic tone. No change has been demonstrated in the ventricular systolic ejection fraction in both types of sleep, and cerebral blood flow increases slightly during REM sleep. With regards to the endocrine functions during sleep, the following have been noted. Prolactin secretion reaches high levels at night during the period of sleep. And as far as TSH secretion, as thyroid stimulating hormone secretion is concerned, now a normal circadian rhythm of TSH secretion has been described, but weak amplitude secretory episodes of TSH occur throughout the sleep-wake cycle. Growth hormone has a secretory peak during NREM sleep, and renin increases in transition from REM sleep or wakefulness to deep NREM sleep and diminishes as sleep becomes lighter or with awakening. Cortisol and ACTH usually have a circadian rhythm with high plasma levels in the morning, which gradually diminish as the day progresses. This rhythm is, however, not modified by sleep per se. Melatonin, as you may be aware of, has a gradual increase in its secretion during the night, starting before the onset of REM sleep. The secretory profile of the reproductive hormones testosterone and luteinizing hormone are independent of the sleep-wake cycle. Now, altered gastrointestinal function during sleep is attributed to varied autonomic nervous system responses. Salivary secretion and the phenomenon of deglutition are particularly reduced during sleep. The resting pressure of the upper esophageal sphincter and the frequency of peristaltic contractions in the body of the esophagus and the lower sphincter seem to diminish during sleep. The duration of the migrating motor complexes appear to be longer and the speed of propagation slower in NREM than REM sleep. Reduced motor, mechanical and electromyographic activity at the colonic level has also been reported. These changes are extremely important to maintain continence during sleep and also facilitate the process of digestion. With regard to the sexual function during sleep, REM sleep is associated with penile erection in males belonging to the physiological age limit. Puberty and old age are two periods in life where nocturnal erections occur as frequently in NREM sleep too. This is penile erection during sleep is attributed to vascular, muscular and hormonal mechanisms. With regards to thermoregulation during sleep, internal temperature evolves according to a circadian rhythm with about minimum occurring at 3 to 4 am and a maximum occurring at 6 pm. An ultradian temperature modulation has also been reported in relation to the sleep-wake cycle with a reduction in temperature during NREM sleep. During sleep, the body temperature changes in function to the surrounding environment, and a sleeping man represents an efficient thermoregulator. The maintenance of our internal temperature is subject to regulatory mechanisms which come into place when the thermoneutral zone is exceeded. The thermoneutral zone is characterized by a zero balance of exchanges between the body and its surrounding. It differs in wakefulness and sleep. At wakefulness, it's 28 degrees Celsius, and at sleep, it's about 30 to 32 degrees Celsius. In the naked man, it also differs according to the sleep stage. The hypothalamus and especially the preoptic region are structures which play a role in thermoregulation. Each sleep state has its own zone of thermoneutrality. The most effective vasoregulation occurs during deep NREM sleep. Cerebral metabolism during sleep was studied using the PET scan, the SPECT and the MRI. During NREM sleep, PET studies showed a net reduction in cerebral activity in the thalamic nuclei, the brainstem, the basal ganglia, the hypothalamus, the basal forebrain, the orbitofrontal and the anterior cingulate cortical regions, the precuneus as well as the right mediotemporal regions. In REM sleep, high activation in the brainstem, particularly in the tegmentum pons, the thalamic and the limbic systems, namely the amygdala, the hippocampus and the cingulate cortex. Intense activity in the limbic system was also found during NREM sleep. So, in summary, NREM sleep has a decrease in cerebral metabolism, while REM sleep there is an increase in cerebral metabolism. Now, let us take a look at the functions of sleep. In the previous eras, the functions of sleep was not very well understood. It is in the 21st century that with the advent of a lot of radio diagnosis, a lot of new functions of sleep are being discovered. So this tabular column gives you a summary of the functions of sleep. The tabular column on the upper part of the screen differentiates the functions between REM sleep and NREM sleep. And the tabular column below talks about the functions which are common to both types of sleep. So here you can see in REM sleep there is strengthening of emotional and procedural memory which takes place. And in NREM sleep you can see there is restoration and energy conservation with the lowering of the BMR, body temperature and energy conservation. Consolidation of declarative memory and cardiovascular health is also maintained in NREM sleep with the lowering of heart rate and blood pressure. In both types of sleep, as far as the homeostasis within the nervous system and toxin clearance is concerned, there is something called the G lymphatic system, which is mediated by the neuroglial cells of the brain, and they get rid of metabolic waste products and prevent neurodegeneration during sleep. There is also promotion of new connections between neurons, a phenomenon known as synaptic plasticity. Sleep is associated with hormone regulation, also contributing to endocrine homeostasis, as we saw previously. Immune surveillance and cytokine protection is also stepped up during sleep, contributing to immunity within the body. And as we conclude this session, I'd like to give you an overview of polysomnography, which is the conclusive diagnostic intervention of sleep, either physiologically or pathologically. And an assessment of sleep is carried out using this technique, and during this technique, the electroencephalogram, the electromyogram and the electroocculogram are recorded. So here you can see a diagrammatic representation of a polysomnography going on, and here you can see the electroocculogram and the electromyogram and the electroencephalogram are recorded. In addition, you have the recording of the respiratory reserve, which is going on this side. Now I'll talk to you about the consequences of sleep deprivation, which has been evidenced by research. This has begun from the year 1988 till date, and they have found out these functional indices have been impaired during sleep. So there's an increase in reaction time, preservation and reduced flexibility, impaired sense of humor, increased risk taking, impaired moral judgment, reduced emotional intelligence, increased negativity with the enhanced memory of adverse events, and increased distractibility. I'm sure we would have gone through some of these indices when we were deprived of sleep. And as I've given you an overview on the basic principles of sleep physiology, I'm sure my colleagues will empower you to a higher level in the subsequent sessions. And I would like to thank the flag bearers of NIPTEL for continuing to give us the opportunity to share our knowledge one and all on this forum. The flag bearers of our institute, Chettinad Hospital and Research Institute, for always motivating us to excel in our endeavors of teaching and research. To all my teachers and students around the world for empowering me with the wisdom of physiology and medicine. And last but not the least, I'd like to thank one and all on this forum, the participants of this forum, for participating in this learning exercise. Thanks to one and all.